Hey guys, in this video we're going to take a look at cell mitosis and a little bit of the cell cycle. So cell mitosis is a process that occurs to divide your normal body cells. It starts off with a couple different cell types, but the first one is what we call interphase. This is a normal cell that's really progressed past a certain point, and it's going to divide. You'll notice that the centrioles start to form, and the centrioles are up here. You can see the microtubule fibers that they have here, these spindle fibers coming off of them. The centrioles are right here, and these centrioles are going to really help in pulling apart the chromosomes, which haven't formed yet, but you can kind of see that the DNA here in interphase is still encased in the nucleus. If we continue, the spindle fibers will form more. This is a prophase cell, and in prophase you'll notice a couple key things happening. First off, in prophase, our nuclear envelope starts to break down. Secondly, we will see chromosomes start to form. So you can see here, chromosomes are labeled. This particular organism has a couple different types of chromosomes. Each chromosome has a centromere at the center of them, and then what happens is these become visible during prophase because they're going to really kind of condense together. Normal cells, the DNA isn't packed this tightly. What it means is that at this point, you can't use that DNA. You just pack them together so that you can transport them. It's the same idea of packing up clothes. You're not able to use clothes that are packed in your bag, but you pack them in the bag so you can easily get them to their location. You will notice that the centrioles have spread since where they were in interphase, and they're moving to opposite poles on the cell. This cell type is prophase. Metaphase is the most distinguishable phase in the entire mitosis process because all of the chromosomes will line up along this imaginary plate called a metaphase plate right along the middle. What actually happens is there's kind of a tug-of-war going on between our spindle fibers, one of which is pulling them to this side, the other of which is pulling them that way, and the tug-of-war ends up pulling them to the middle. In mitosis, they're all lined up in the middle, and it's very, very easy to identify. At this point in time, our nuclear envelope is completely gone, and they're all lined up in the middle. Metaphase cell. As we progress, that tug-of-war will eventually cause the centromere to be pulled apart in those chromosomes. And what actually happens is not that it's a true tug-of-war where they're pulling in opposite, like these are ropes being pulled this direction. Instead, on this, what remains of the centromere, there's actually a thing called a kinetochore. And what the kinetochore does is it walks along the spindle fiber towards your centrioles, and the spindle fibers that were here are actually broken down. Either way, the bottom line is these chromosomes are pulled to each end, and they're pulled apart in anaphase. Anaphase cells are pretty easy to identify because they are split chromosomes that are no longer found in the middle and are being pulled towards the edges. If we continue to get to telophase, a couple key things will be noted in this late telophase. Your nuclear envelope is reforming here. And you'll notice that there isn't one nuclear envelope, there are two. And that's because at this point, we really are at about two different cells in this late telophase. You will also start to see the beginning of the next process we'll talk about here, where these cells are kind of being pushed together. That right now tells me that I know this is an animal cell, because that isn't how cytokinesis happens in plants. But in late telophase, everything else is going to be split too. All of your organelles are going to move to one side or the other as your cell becomes two separate cells. This actually shows you cytokinesis, and here you have a microscope view of it, and then an artist's rendition. The thing you'll notice about cytokinesis is this is an animal cell, and you have this cleavage furrow form here. And what that cleavage furrow is it's your actual cytoskeleton kind of pinching itself in. Were this a plant cell, you wouldn't have this cleavage furrow. Instead, plant cells would just form what they call a cell wall right in between here, and they'll just build a new cell wall there. They don't have to pinch in. So it is different in plant cells and animal cells, but cytokinesis actually splits the cytoplasm. That leads us to our entire process of the cell cycle. You'll notice that what we talked about was mitosis, and mitosis is found in this one little section right here. And then we have cytokinesis, that is the splitting of the cytoplasm. All of that happens very fast. It's cell division. The vast majority of your cell's life is actually spent in interphase, which consists of the G1 or growth phase. That's where cells just grow normally. They're going to expand in size. They reach a certain point, and if they get a growth factor, which tells them, hey, you're going to need to divide, they're going to go through the S phase, which is where they copy their DNA. So in the S phase, they're going to replicate their DNA, copy it so they have 2 copies. And you might wonder, why in the world do they need 2 copies of the DNA? Well, every cell needs its own copy, so it has instructions for what to do. So before it splits, it's got to copy it. After the S phase, which is synthesis or cell replication, it's going to move into G2, which is kind of a preparation for mitosis. There are a lot of checkpoints that happen in G2, and if those aren't achieved properly, if something goes wrong, the cell will stop right here in G2 and it won't go anywhere. In fact, sometimes it'll be subject to apoptosis or programmed cell death. But G2 is another like growth type phase preparing for mitosis. You'll notice that G1, S, and G2 are much longer within the scale of time than our cell division phase. So here I have a picture of a broken bone that is mending itself. And these new bone cells are generating as part of the process of mitosis. They're receiving a growth factor that says, look, you need to divide. So they are dividing. They're actually generating new cells there. Anytime you have an injury or anytime you grow, mitosis is the cause of that. Because what mitosis is one cell becoming 2. That could happen because there's a cut or broken bone like here, or it could happen because you are growing as a person.